Hello. Okay, so I guess we'll just do it old school. Sorry about that. But also thank you for letting me do my little dance break because I really appreciated that. I am so thankful for every single one of you for coming out and helping me celebrate all of the work I've accomplished at UC Irvine. And I know that I wouldn't be on this stage and being able to present the work that I did without everyone's support here. So without further ado, I'm so excited to share with you all about the acidic adventures that I had with secondary organic aerosols. And so my passion for air quality and my passion for atmospheric chemistry actually stemmed from my family. My family grew up in a southeastern province in China, Guangdong, China, and they eventually decided to immigrate to the United States because they wanted more opportunities for their family. And so when they decided to move to the United States, they chose Los Angeles. And as you can see here in both these images, in the Guangdong province and LA, the air quality is pretty bad. And watching my family kind of experience all the health effects due to air quality was kind of hard. And so that's why I chose to work in this industry or work in this subfield, because I really wanted to create equitable air quality for everyone, especially my family. And so as you see in these images, we see atmospheric aerosols. And atmospheric aerosols are solid or liquid particles suspended in air. They're really important for us to study because they not only affect our visibility, they can also impact climate by interacting with solar radiation. And they also can indirectly affect climate due to their interferences with cloud properties. And finally, once we breathe all of these aerosols in, they can cause a public health crisis because of the struggles. Once you breathe aerosol particles in, they can cause a variety of respiratory illnesses. And so there are two classifications on aerosols. There are primary aerosols, and that is emitted directly into the atmosphere, and they have a variety of sources, including dust storms, oceans, industry, and vegetation. But there's also another classification of aerosols, and that is secondary organic aerosols. And these are formed in the atmosphere via chemical reactions. This includes secondary inorganic aerosols, which includes inorganic constituents. However, they also have organic aerosol, which is the primary focus of my work. And so how these secondary organic aerosols, or SOA, is formed is that gas emissions are emitted from a variety of these sources, and they emit something called volatile organic compounds. These volatile organic compounds then get oxidized and coagulate into organic aerosols. And so these are really important, and they're very complex, and they have a variety of properties. And so we also know that aerosols can be in transport. So once they are emitted or formed, they can transport all over the world and experience different environmental conditions or species. And so they can last in the atmosphere upwards to days and weeks. Additionally, when they experience new environmental conditions or species, that is called chemical aging. And so that is what is kind of the main focus of the work that I'm doing on aerosols. We also know that aerosols can have a wide range of acidity, and so this graph shows you the different acidities in the atmosphere. The red corresponds to cloud water, where the blue corresponds to fine particles. These lines indicate different models that we use, and these markers indicate the observations within the field. And you can see that cloud water tends to be a little bit more basic, have a pH around 8, and then goes all the way down to pH 3, where aerosols have a little bit more acidity to them, and they start around pH 6, and then goes all the way down to negative pHs. As you can see in these graphs, the lines and the markers don't necessarily line up. They are not perfect, and so there's a discrepancy between field observations and models. And so that's where I come in with the experimental or lab experiments. I want to help help make this match a little bit better. And so composition can actually play a large role in the acidity of aerosols, as well as their source and ambient relative humidity. And we see here in this image that sea spray aerosols and dust aerosols tend to be a little bit more basic due to their cation, or their addition of cations and ions present. In addition, biomass burning and sulfate organics tend to be a little bit more acidic because of the presence of more acids and organic acids. And so we know that acidity, or chemical composition, can play a large role in the acidity of aerosols, but thereby acidity can then play a large role in the chemical composition. And so understanding these two properties and the relationship between the two is really important for us to understand the whole picture of why aerosols are important. And like I said, chemical composition is important for multiple factors. First, they can control the fate and transport of aerosols. They can have direct and indirect effects. For example, light-absorbing compound can directly absorb solar radiation, causing a warming effect. And then they also can kind of change cloud properties due to their ability to act as cloud condensation nuclei. And finally, the chemical composition can also determine the extent of which aerosols can impact our health. And so, all in all, chemical compositions is huge, so that it's huge to understand the whole picture of why aerosols are cool. And so there has been a huge amount of work being done on this relationship between acidity and composition. First, we have chamber studies that have shown enhanced particle production in the presence of acidic seeds. Studies have also shown the reactive uptakes of individual products of oxidation of VOCs onto acidified particles. There also have been reactive uptakes of aldehydes into sulfuric acid bulk solutions, forming light-absorbing compounds. And finally, in our group recently, we saw that the evaporation of bulk SOA can actually cause significant changes in the SOA composition, forming organosulfur formation. And so, although there has been a huge push to understand the relationship between acidity and composition, there is still large uncertainty pertaining to the role of acidity in aerosol aging specifically. And so that leads me to my overarching question what drives my thesis, is how does acidic environments impact aerosol aging processes and composition over long-term aging? And so this is really important for two things. One, we're able to elucidate possible aging mechanisms and kinetics over organic aerosols in different environmental conditions. And two, we get to know a little bit more about the upper troposphere and lower stratosphere and how organic aerosols are in that realm. And this is a very unique area because the upper troposphere and lower stratosphere is very acidic, it is cold, it is dry, and anything that gets uplifted into the upper troposphere or lower stratosphere tends to have a longer lifetime up there. And so the first thing I wanted to look at is alpha-pinene ozonolysis, secondary organic aerosols, and how it interacts with a variety of concentration of sulfuric acid. And why I chose alpha-pinene ozonolysis is because it's a prominent biogenic gas that gets oxidized in the atmosphere. So if we know more about alpha-pinene ozonolysis, we know a little bit more about the organic aerosols as a whole. And so I wanted to look at the chemical composition and their optical properties. And so in order to do this, I first formed the secondary organic aerosols, and we formed them using an oxidative flow reactor. And so briefly how this works is that we have oxygen flowing into a line that gets generated to ozone, and then that flows into the flow tube. At the same time, we have clean air flowing into the system and picking up our VOC of interest. In the flow tube, the alpha-pinene and the ozone mix, forming secondary organic aerosols, and then we collect them using on a foil substrate. And so after I formed my secondary organic aerosols, I then took these substrates and divided it into three. I first dissolved it using acetonitrile, rotovapped all of the acetonitrile away, and then added water. And this kind of acts as our control. What happens if there's no acid present? Next, I did the same experimental method to a second fraction, but this time I added sulfuric acid in it and aged it for 2 days. We then took these samples and then analyzed it using a high pressure liquid chromatography, photodiode array, high resolution mass spectrometry. And you probably was like, oh my God, that's a very long acronym. And then you're right, but to simplify it, it basically helps separate our compounds and helps us analyze the absorbance and the composition of each separated compound. In the next fraction, we did the same experimental procedure, but we did something a little bit different, in which we added our sulfuric acid and then immediately put it into our UV vis. We analyzed it using UV vis for 2 days, and that allows us to understand the rate of chromophore formation. And so in doing this, we kind of know more about this relationship between alpha-pinene, SOA, and acidity. So let's get into data. So here I show you the mass spectra of fresh alpha-pinene SOA, and then alpha-pinene SOA aged in water, in pH 3 and in pH 2. And there are 2 parts of this mass spectra I want you to focus on. There is a monomer region, which accounts for smaller molecules, and then there's a dimer region, and that accounts for more complex molecules. And as you can see here in all of the mass spectra, each of them look very similar to each other. There's not much deviation between the monomer region and the dimer region. And so that led us to conclude there's a lot of similarities. So in order to push this forward, we looked at the composition as a whole, right? So here we have the relative intensity of the different types of compounds in our system. The green represents compounds with carbon, hydrogen, and oxygen, and the red accounts for compounds with carbon, hydrogen, oxygen, and sulfur. And as you can see in all of these, there's not much change. Majority of the sample is CHO compounds. It's all green. And that led us to believe that there's no real evidence of acid-catalyzed aging occurring under moderately acidic conditions. And so then we decided, okay, let's push this forward again. What if we drove this system and just added a bunch of acid to it? And so here we have fresh alpha-pinene SOA, the mass spectra, and we compared it against the SOA aged in pH 1, pH 0, and pH -1 conditions. And we see here that for the first 3 samples that I mentioned, there is that monomer region, and then there's that dimer region, indicating there's not much change happening. And it's very similar. However, I do want to bring your attention to this lower plot here, where we see a change in peak pattern when we age the SOA in pH -1 conditions. There's not much dimer region, there's not much monomer region. It kind of just like, okay, here are my peaks. And there are also new peaks that are formed too. So if we look at the relative abundance for each of these samples, we see that as we age our SOA in aged our SOA in pH -1 conditions, we see an increase in the amount of organosulfur compounds, an increase in the red trace, and it accounts for roughly 30% of the sample. And so that has led us to believe that aging aerosols in highly concentrated sulfuric acid leads to organosulfur formation. But another thing that was really interesting was that when we ran these samples, there is a color change that occurs. And so we see that a visual representation of these samples over 5, 24 hours, and 48 hours. And so there's a deep change in the color of the sample. And so that's where we decided to analyze the sample using UV vis spectroscopy. So we analyzed it, and we were able to see a growth increase over time. So the top absorption spectra indicates your alpha-pinene aged in 5.6 molar sulfuric acid, which is roughly -0.86, whereas 10 molar sulfuric acid represents roughly pH 1. And so for the sample up here, we see 2 peaks increasing gradually over time, whereas this bottom peak over here, or this bottom graph over here, we see occurrence of more peaks. But there was a little bit different in terms of rates happening. So to take a closer look, we monitor the peak as a function of time for every single peak. And we saw that for the less acidic samples, that the growth of these increase as a function of time, but it's gradual. However, for some of the peaks in our pH -1, some peaks increased rapidly and then plateaued, whereas some peaks increased over time slowly. And so this kind of rate forms really quickly. It forms quickly around 3 hours and then starts to plateau. And so these are differences between the 2 samples that were really interesting to see. And so I was able to see that light-absorbing compounds formed in highly concentrated sulfuric acid, and they formed, but they were forming at different rates. And so to take a closer look at the light-absorbing compounds that we had, we wanted to look at the chromatograms. And these chromatograms is the separation of compounds that absorb light, and then the separation of compounds that have a mass to charge of 151. And we see in both of these samples, both of these chromatograms, we have that triplet of peaks occurring, indicating that maybe this sample right here, 151, is the cause of light absorption. However, due to the complexity of secondary organic aerosols and the range of compounds that occur, it was really difficult to determine exactly what the structure of chromophores are. And so that kind of left us with this lingering question for this specific project. And so in conclusion for this first part of my talk, we aged alpha-pinene secondary organic aerosols with a range of sulfuric acid, and we found that under moderately acidic conditions, there's no real evidence of acid-catalyzed processes. However, when we started to hammer a bunch of sulfuric acid into our sample, it leads to acid-catalyzed reactions resulting in organosulfur formation. And finally, we were able to find that light-absorbing compounds formed in the same conditions where it's highly acidic. However, there's still that lingering question of what these compounds are, and it was kind of frustrating. So then, you know what, I was all like, let's just simplify the system. If our system was a little too complex, let's simplify it. And that led to this question right here, and the second part of my talk, where I looked at the change in chemical composition and optical properties of the monoterpene oxidation product in acid. So basically, I chose 2 compounds in the alpha-pinene SOA, and specifically, I chose panonic acid and pinoaldehyde. So panonic acid is at the top, and it has a ketone, it has a cyclobutane ring, and it has a carboxylic acid. The one on the bottom, which shows pinoaldehyde, it has the same ketone, the same cyclobutane ring. However, this time it has an aldehyde. And so we chose these specific compounds because they were abundant and have been seen in alpha-pinene SOA. And I did the same experiment where I took an aliquot of the pure sample, aged it in a variety of sulfuric acid, aged it for 2 days, and did 3 types of analysis on it. First, I did NMR analysis, and that allows us to determine more about chemical structure. Next, I did UV-vis spectroscopy. And again, like I said previously, that allows us to determine the rate of formation of our sample, or rate of formation of the chromophores that are present. And finally, I analyzed it using this big, long acronym again. And just to simplify, it allows us to separate this different compounds and then analyze its absorbance and composition of it. And so I first want to start off with panonic acid. Again, remember, panonic acid has that carboxylic acid, and within 24 hours, I was a little bit disappointed. There's no color change, and I was heartbroken, and it felt like it was a failure on my end. However, when I started looking at the chromatograms, the total ion chromatograms, it told a whole different story. First, we have two different peaks. So this is the total ion chromatogram of our sample aged in water, pH 3, pH negative 0.86, and pH negative 1. And so we see the occurrence of two peaks here. This first peak right here that is roughly around elutes around 9 minutes, that's actually our starting material. That's panonic acid. However, when we start decreasing or increasing the acidity of which we age the system, there's a second peak that occurs, and that peak occurs a little bit earlier. And so what we did was like, okay, so we have a change in the total ion chromatogram. Do we have a change in the UV vis? So here I show you the UV vis of the panonic acid aged in pH 3 and aged in pH negative 1. And we see that the sample aged in pH 3 doesn't really have a change. It's just the same line all over again, time after time. However, when we aged it in pH negative 1, we see the two peaks decrease as a function of time. And this is associated with the N to pi star and pi to pi star transitions, indicating that there is a chemical change. So at least for panonic acid, we're able to see that it might not have a change in color, but it does have a change in chemical structure, which is really exciting. And so after countless research in literature, we actually found a paper that kind of shows us what's happening. And it basically says that panonic acid undergoes an enol ketotautomerization and then rearranges to form a compound called homoterpenoid methyl ketone. And we were able to confirm this using NMR spectroscopy too. And so we were able to say confidently that panonic acid forms this homoterpenoid methyl ketone under highly acidic conditions. So let's move on to the star of the show, phenolaldehyde. And as you can see, phenolaldehyde has the aldehyde instead of a carboxylic acid. And within a first couple of minutes, my heart skipped a beat. I was so incredibly happy to see this color change because I'm like, oh my gosh, this might be the answer. So if we take a look at the total ion chromatogram, I want to kind of walk you step by step. So we have phenolaldehyde aged in water, aged in pH 1, aged in pH 0, and then aged in pH negative 1. And so this first peak is associated with panonic acid. It's in our starting material. It was really difficult to fully purify phenolaldehyde, and so we have a little bit of panonic acid. And for the pH negative 1 sample, that peak is slightly shifted because of the acid catalyzed reactions we've seen before, forming homoterpenoid methyl ketone. The second broad peak that elutes out around 9 minutes is actually our phenolaldehyde. It's our starting material. And as you can see here, there are 3 samples with that peak. Water, pH 1, and pH negative 1. However, in pH negative 1, we see that peak completely disappear, indicating that this reactant had fully reacted away. And finally, we have this same triplet of peaks that occur at pH negative 1. And this doesn't happen at any other pHs except pH negative 1. And it's that haunting triplet of peaks again. However, even though I know this triplet of peak is because of phenolaldehyde, I still don't know what this structure is. And this was really frustrating. And so we kind of did that same analysis. So we were able to conclude, at least for phenolaldehyde, there is a color change and a change in chemical composition. And so in order to extend my understanding of this reaction, I looked at the different types of chromatograms. First, I looked at the total ion chromatogram, and again, that shows you the separation of compound in our reaction mixture. Next, I looked at the PDA chromatogram, and that PDA chromatogram shows us the separation of compounds that absorb light. And finally, I looked at the single ion chromatogram for specifically that have a mass to charge of 151. And for every single one of them, there is that haunting triplet of peaks again, indicating that our compound that has a mass to charge 151 and that has this molecular formula is our light-absorbing compound and is the product from phenolaldehyde. We then took a closer look using UV-vis spectroscopy and monitored it as a function of time. And we see here two absorption spectra for pH negative 0.86 and pH negative 1. And they look strikingly similar to the data that I've collected prior, where we have two peaks that increase as there's a gradual growth over time. And for this pH negative 1 sample, we have a huge increase, peak around 370, and there's a variety of peaks. There are shoulders, there are a huge peak, there's a small peak here, kind of thing. So it looks very strikingly similar to the sample that I've collected previously. However, there is still the question of what is this compound? And this was the bane of my existence for the last couple of months. So in order to do this, I actually was in discussion with a organic chemistry group here at UC Irvine, the Rychnovsky group, and was able to collaborate with them. After many discussions, we decided to upscale the reaction. So we upscaled the reaction by roughly 500 so that we can generate enough material for NMR spectroscopy. Our collaborators, the Rychnovsky group, was able to separate and purify the sample. And then Jess, she was able to run NMR spectroscopy on it. And again, that allows us to learn more about the chemical structure and where things are. And so we separated it out and found that there are three things that prominently occurs. And these are the beautiful molecules that Jess figured out. And this first molecule right here, you probably was like, oh, that's a little bit familiar, right? That's actually homoterpenoid methyl ketone. And that is because when you oxidize phenolaldehyde, you're gonna get the carboxylic acid derivative of it, or the carboxylic acid of the aldehyde of it, I guess. And then that leads to the formation of homoterpenoid methyl ketone under highly acidic conditions. However, these two compounds, they're different. And that got me really excited because this could be our light-absorbing compounds that we haven't seen before. So let's talk about these reactions one by one. So we know that phenolaldehyde under highly acidic conditions form this compound of interest that has a 5-member ring with two alkenes, one in the ring and one outside of the ring. And after many discussions with the Rychnovsky group, we were able to determine that it undergoes a couple of steps in order to form. And as a non-organic chemistry person, arrow pushing scares me so much. And so I was able to simplify the reaction into these 5 key steps. First, with the phenolaldehyde, we protonate the ketone. It follows by a ring opening of this cyclobutane, followed by a hydride shift. And so from that hydride shift, we get this intermediate. And so in that intermediate, we're then able to protonate the aldehyde, which then leads to an aldyl condensation, forming a carbon-carbon bond to form this 5-member ring right here. And so this 5-member ring has this IUPAC name, and I do not want to say the IUPAC name because it's too long. But we've actually seen this terpene oxidation product. However, it's been seen as a derivative or as it's derived from alpha terpenoid, but it hasn't been seen in alpha pinene oxidation products. So that was really cool. And so if we look back at my haunting triple of peaks, we see that this compound right here is associated with this latter peak that elutes out around 12 minutes. And so we now know that at least this compound right here is associated with this peak, but what about this orange one? What about it? So we're able to isolate at least one of them, and we see that it forms this compound. And it forms a very similar compound as the minor product. However, it has 2 alkenes in the 5-member ring rather than 1 inside and 1 outside. And so it kind of undergoes the same transitions or the same reaction as the previous one, where there is a protonation of the ketone, a ring opening of the cyclobutane, and then a hydride shift. That hydride shift creates this intermediate, and this intermediate then undergoes a protonation of the aldehyde and aldyl condensation to form this product right here. And so this product is this name. If we look closer at it, this is the major product of this peak, this orange trace right here. And now you might be thinking, well, there's 2 peaks in that orange trace. Yeah, that is correct. There's 2 peaks. The unfortunate thing is that this smaller peak, it was really difficult to isolate because it was in trace amounts. However, we know that it's a very similar compound to this main larger peak right here. So we predicted, okay, maybe it's this second compound right here that's slightly different, but kind of the same. So that's why it lutes out at the same time, and it was very difficult to separate and isolate. So it probably forms 2 regioisomers of the product. And this compound we actually have not seen in literature before. And so, at least for this project, I was able to look at the impacts of highly acidic conditions on pinonic acid and phenolaldehyde. We were able to confirm that pinonic acid was able to rearrange into homoterpenoid methyl ketone under acidic conditions, while phenolaldehyde was able to form 3 chromophores under acidic conditions. This is really important because now we know that these compounds absorb light, which then can eventually be converted into heat and then cause a warming effect on Earth. And so you might be wondering what happens next. Well, in any story nowadays, there's always new adventures to be sung and new adventures to be had. And so now these stories have a prequel. And so in the first project that I did, I actually did it in everything in the absence of acidity. And so we looked at how water and different forms of water can play a role into long-term aging of these biogenic monoterpenes. And so there has been an increasing amount of literature that looked at SOA formation and short-term aging mechanism and how water can affect that. However, there hasn't been much about long-term SOA aging. And so we ran a bunch of samples from biogenic monoterpenes, aged in liquid water and water vapor, and we monitored the mass spec of it. And so we basically see the SOA, the composition of the SOA changed during long-term aging. However, the extent of the change is relatively small because we see here there's that same monomer region, the same dimer region, and the same peaks occur, probably at like very variable intensities or slight variations in intensity. And so this allowed us to conclude that water is not really a strong driver in long-term aging processes. And so, if this is the prequel to my story, what happens when there's a sequel? Well, we monitored the biogenic and anthropogenic secondary organic aerosols, and we looked at how acid can affect the fluorescence of these compounds. And this is really important because fluorescence is often used to characterize primary biological aerosol particles. And so if acid can create fluorescent compounds, this may cause a lot of interferences with the fluorophores in these bioaerosols. And so what we did was we analyzed the fluorescence of kind of a lot of types of secondary organic aerosols with a variety of VOC precursor and oxidants. And we found that it was only until we aged these samples in pH -1 in which fluorescence starts to occur. And so we see here on these graphs that they form two types of fluorophores, highly oxygenated fluorophores and less oxygenated fluorophores, which are denoted in green and pink boxes respectively. We also then took it a little further and started to look at the dilution of it, so the impact of water on these fluorophores. So we took these samples, we diluted it with a variety of amounts of water and see how the absorbance changed and the fluorescence changed as we added water. And so we see here on this graph that the biogenic mass spectra, as you add water, it starts to shift. The mass spectra starts to change, and especially for that biogenic the biogenic SOA. However, for toluene and xylene, which are associated with anthropogenic SOA, it only changes slightly. And so we know that the shapes of this changes, but then we looked closer at the fluorescence of it, and that same change that occurs in absorbance doesn't happen in fluorescence. So that could indicate that the fluorophores and chromophores are different molecules or different species. And so overall, this project led us to conclude that acidity is a major driver in the formation of fluorophores. And this can pose a huge challenge for measuring and analyzing primary biological aerosol particles. Okay, and so if you all have not been listening, that's totally fine. There are three main things I want you to take away. One is that acidity of aerosol is an important property that plays a crucial role in the physical and chemical processes in the atmosphere. Two, that the laboratory studies presented here allows us to see a molecular understanding of the synergistic effects between monoterpene oxidation products that are from biological sources and other compounds of anthropogenic origin like sulfuric acid. And the one thing I really do want to drive home is that there is a change in the chemical composition and optical properties, but it doesn't really occur until we age these organic aerosols in highly acidic conditions. And so my hope is that with the research that I've collected here, along with future field studies and atmospheric modeling, we can have a more fundamental understanding of the atmosphere that could eventually lead to global and local environmental policies and further improve our air quality and sustainability. Thank you. So I guess this is the fun part of the acknowledgements. So one little birdie told me never to forget this moment. So I am going to take a quick photo, real quick. So say hi, everyone. Hi. Thank you. But I really wanted to do that. I just don't want to forget this, at least this moment being up here on this stage. The first thing I do want to do is thank my advisor, Surya Niskordoff. Thank you for giving me the liberty and freedom to pursue as many and as weird extracurricular activities that I want. That's besides chemistry. Your support in all of my extracurricular activities and the projects has been truly, truly a confidence booster. And I appreciate your dedication to, and your knowledge of chemistry. Wow, that is, your brain is crazy. I do want to thank Anne-Marie and Jim. You two have been such a great pillars of Air UCI. You two have taught a lot of us, whether it be about modeling, whether it be about instrumentation, but you two inspire us to be really awesome atmospheric chemists. And I'm really thankful to have you two as role models and to at least be associated with y'all. I want to thank my group, past and present. Thank you so much for giving me a space to bounce ideas off of, to fail, to learn about troubleshooting instrumentation. I truly am inspired by every single one of you all, and you all make me a better researcher and a better scientist. I also just really want to highlight my undergrads, Daniel Vite, Lucia Liu, and Jet Wong. Thank you so much for letting me be your mentor, your graduate mentor. It has been a pleasure to watch you grow as a scientist. Your dedication and your hard work to your respective projects is astounding, and I hope you learned I hope you learned a lot, because I learned a lot from you. I want to thank the Carleton group. They have been I've been an honorary member of the Carleton group because I keep barging into the office and keep having a lot of mental breakdowns there. So I just want to thank you for giving me a space to have a new perspective on research and in life. You all deal with a lot of me barging in. I want to thank AirUCI, but specifically I want to thank Lisa Winning and Vero. Man, you guys are two of the backbones of AirUCI. We as grad students would not be here without your discussion, without your dedication to us, and without the work that you all to teach us. Thank you for letting me just talk rambles about chemistry to you. I really appreciate it. I also want to just have a quick shout out to all of the other AirUCI groups. I am so grateful to be in a community of scientists, that has an amazing, an amazing brain collectively. I think you all are just wonderful, and I'm so glad that we are able to go through grad school together. I want to thank my collaborators, Jess and Scott. I know Jess was here earlier. I did not know a lot about organic chemistry going into grad school, so without them, my organic chemistry knowledge would still be very slim. I want to thank my UCSD mentors, Vicky Gracian, Ellen Cottons, Leora Mail, and the rest of the Gracian group. They saw me when I was a very small, very naive undergraduate researcher, and they were able to help me grow to this confident scientist that I am today. I just want to have a special shout out to Ellen. I remember when I was going into UC Irvine, I told her I was really scared to do research because up until that point, I've never done research without Ellen by my side. I would always bug her, I would always ask stupid questions, I would always just ask her to use her computer. But even though she was never here physically at UC Irvine, she was always cheering me on from afar. And she was always there by my side figuratively. And so I can confidently say to Ellen that I've always done research when she was right beside me. I do want to give a special mention to a couple of things. Barkeeper's friend, I do a lot of acid work, and I have destroyed or temporarily destroyed the cone and corroded the cone with my sulfuric acid. So shout out to Barkeeper's friend for allowing me to keep that shiny stainless steel. Also, Arm & Hammer baking soda allows me to is a cheap and effective way to neutralize my acid. So big shout out to them. I also want to thank the rest of my cohort, whether it be big or small. Thank you for just those conversations, those insights to chemistry and life. I'm glad we made it. And finally, I want to thank my funding sources. I would not be able to do all this cool chemistry without them. And the facilities, they are absolutely bonkers for giving me money. But yeah, and so there are 3 other additional groups I want to thank. Ridge to Reef. It's a community of environmental scientists, and it extends beyond chemistry, and so they were able to cultivate a renewed passion in environmental justice in me, and I'm so thankful for them. Without them, I would not be able to intern at a company that I have so dear to my heart, IQR North America. Working with these coworkers has truly been inspirational and has been such a blessing to work with. And so I also really want to just give a heartfelt thank you to Dr. Christy Chester-Shorter. She is an incredible role model, and she works tirelessly to make our team runs, and her dedication and how her mind works is beyond me, and I'm so thankful to have her as a role model. I also want to thank Community. I developed this program with a couple of my peers, and to see it flourish into what it is now and to work with any of the mentors and mentees within this program has truly been inspirational, and thank you for letting me have my vision into a reality. There are a lot of friends I've met along the way, and they are amazing. I just want to say that I would not be here without any of you, and I'm so thankful for every single one of you. I especially want to thank my roommate, Mariana. Thank you for challenging me, and thank you for making me believe more in myself than ever. I want to have a huge shout out to nature. Going on to camping trips has been a blessing, and it always gives me a new perspective. And going on these adventures with these hooligans has been an adventure of a lifetime. And for any of the current graduate students, remember to take a break, because nature reminded me to take a break. And then to all my friends outside of grad school, you all have given me so much perspective outside of academia, and to remind myself that I am more than a graduate student. Thank you. I do want to say a special thank you to my partner, Eduardo. Thank you for always being there for me, cooking for me, chauffeuring me around, and always encouraging me. I'm excited to start our next chapter of our lives together. And finally, I want to dedicate my entire PhD to my family. They are the reason why I'm here. I love them so much. They're not here currently, but they're online, so I just want to say hi. I love them with so much, and the amount that they sacrifice for me, and the opportunities that they have afforded me has been truly inspirational. Thank you for being my parents. My dad is no longer with us today, but I know that he would be really proud of me and all of the things I've done. So you may not, okay, so some of you all know, but I've talked a lot about adventures. I've talked a lot about the acidic adventures of secondary organic aerosols, but what's next for me? I'm so excited to share with you that I actually took a position with the California Air Resources Board, and I get to be an air pollution specialist for them, and I'm so excited for the work that is to come with them, and I actually start this Monday. And with that, thank you for all of the support throughout the years. Thank you for showing up and being here for me, and I'm so happy to take any questions. Are there any questions?
You have one online.
I have one online, okay.
Why was foil chosen as the collections media? Do you have reason to believe it didn't affect the composition of the aerosol?
Yeah, so I chose foil. Well, I chose foils because I used a microorifice uniform deposit impactor, and that allowed us to have a uniform film on our substrate, and so for that I can have an even amount of SOA for the aerosols that I collect. So yes, it could. I don't think foil can have a huge impact on the composition initially, because it never was truly once we collected it and once we extracted the SOA from it, that's all we really used for it. Jen.
I was wondering if you did a comparison between long-term exposure with a less acidic versus short exposure with something of high.
So no, I did not do that, but I did think about that. I think that would be a great future directions for this project, to see if there is eventually, does it even lead to these 5-member rings of phenolaldehyde? Does it even lead to like light-absorbing compounds? So that wasn't necessarily the focus of my work, but I definitely think that should be an area that should be explored in the future to see if there's acidic reactions over long term, especially since in the upper troposphere and lower stratosphere, organic aerosols can actually just stay up there for years if there's no deposition. On?
Do you see similar reactivity if you use like a different acid? Repeat the question.
So the question is, do I see similar reactivity if I change the acid? So I actually haven't tried that. I mainly looked at sulfuric acid because that is one of the more abundant acids in the atmosphere, but I think it is a good idea to try nitric acid, if anything, but it's not as abundant in comparison to sulfuric acid. Alyssa?
Hi, thank you. First of all, excellent talk. I'm really honored to be here. My question is, have you ever been able to detect the same transformations of aldehyde in more complex mixtures, or if you haven't looked at that yet, do you predict that you'd still see that formation of homoterpenyl methyl ketone?
So for penonic acid, you say? Or for penol?
Yeah, phenolaldehyde, penonic acid. Do you predict that the same transformations would occur if you have that really low pH, but in a more complex mixture?
It honestly can go either way, but I think because SOA is very complex and it has a lot of different organic molecules in it, it could be that penonic acid, that ring breaks, and then it can be reactive towards another product in the system.
Wonderful talk. I think Scott Ruknovsky is very happy for you providing those mechanism problems for when he teaches that class. It's a little complicated enough for an exam. I'm curious if you can comment on, I'm guessing with an increase in CO2 and like our environment, the environment in general is getting more acidic. So does that mean that overall all of these sort of molecules that exist in space are becoming more toxic? Is there an issue with increasing acidity in the environment for what that means for us?
So that's a great question. The question is, with an increase in acidity in our atmosphere, are these compounds, do they pose any toxicity to us? I would say it really depends. I'll give you a diplomatic answer. I think it really depends. Acidity is really bad for us because it can cause a lot of, acid rain has been a huge problem in a previous lifetime, I guess. But in order to understand the toxicity of it, you would have to do a more comprehensive study on these molecules. And look more about and work with the School of Medicine and to see and the public health factor to see how toxic they are. They could be because if you breathe them in, oxygen can do. Sorry, Jonas, you had a question.
Yeah. So on your the early part of your talk, you were talking about the effect of the sulfuric acid on your mass spec and stuff, and you showed an increase in sulfur-containing compounds. But the mechanisms with the specific compounds you didn't add any sulfur, so I was just curious if you had ideas where that sulfur would add in a different mechanism or something.
So within the mechanism.
Or a different mechanism or...
Yeah, I think that the ring opening of the cyclobutane can really initiate any reaction to occur because now you have a free aldehyde and you have an alcohol occurring with the double bond. So yes, it could. That is a really interesting perspective to approach the question on because you can definitely try. I would look at the common organosulfurs and see if they have common similarities to the 5-member ring that I proposed. But because there's not, because the reaction doesn't really pose a lot of sulfur in it, it could just be very different. Hi. Thank you so much. We will now proceed to the closed portion of our talk with my committee members.